you can train, eat properly, and still have low testosterone. Because testosterone is not just a hormone, it is the result of a complex regulatory system, and this system primarily responds not to supplements but to signals of survival, stress, and energy. Testosterone is regulated through the HPG axis. The hypothalamus releases GnRH, the pituitary releases LH, Leydig cells in the testes produce testosterone. But in parallel, there is another system, the HPA axis. Hypothalamus, pituitary, adrenal glands, cortisol, and it is this system that most often suppresses testosterone. Sleep in 2011, Leppold and Van Cotter, University of Chicago, showed sleep restriction to 5 hours per night for 1 week reduces testosterone levels by 10 to 15%. Mechanism: testosterone is produced in pulses, and its peak occurs during deep sleep during sleep. GnRH secretion increases LH, increases Leydig cells are activated with sleep deprivation. The amplitude of GnRH pulses decreases. LH decreases, testicular stimulation weakens additionally. Evening cortisol increases, which suppresses the hypothalamus as a result. Double suppression through sleep and through stress. Psychology and social status. Testosterone is closely linked to the brain's reward system. In 1992, Alan Mazur showed that testosterone levels correlate with social status. In 1999, Mazur and Booth described the winner effect after a win. Dopamine increases the hypothalamus, increases GnRH, LH, increases testosterone rises, but the reverse mechanism is more important. After failure, dopamine decreases the amygdala, threat center, is activated the HPA axis, is activated cortisol, increases cortisol, suppresses the hypothalamus, reduces GnRH, reduces LH, reduces testosterone. In a study by Bernhardt et al., 1998, it was shown, even in football fans, testosterone levels change depending on the match outcome. This means the brain reacts not only to real events, but also to the perception of winning or losing. Chronic failure maintains high cortisol, suppresses the HPG axis, and leads to persistently low testosterone. Sugar and carbohydrate load. In 2013, Coronia et al. Showed ingestion of 75 g of glucose reduces testosterone by about 25% in healthy men. The drop begins within 20 to 30 minutes and reaches its lowest point at 60 to 120 minutes. Mechanisms: rapid spike in glucose effects on central hormonal regulation, possible direct effect on Leydig cells. Additionally, with regular sugar consumption, insulin resistance develops, insulin levels increase, inflammation increases. This leads to reduced tissue sensitivity, impaired hormonal regulation, and decreased testosterone. Body fat and aromatization. Adipose tissue is an active endocrine organ. In endocrine reviews and studies by Travasin et al., it is shown the higher the body fat percentage, the lower the testosterone level. The main mechanism is aromatization. The enzyme aromatase converts testosterone into estradiol with increased fat. Aromatase activity increases estradiol levels, increase estradiol, suppresses the hypothalamus, reduces GnRH, reduces LH, reduces testosterone. Additionally, fat tissue releases cytokines, inflammatory molecules, which disrupt hypothalamic function, reduce receptor sensitivity, increase hormonal imbalance. This creates a vicious cycle. More fat, less testosterone, even more fat. Chronic stress is one of the most powerful factors. In studies by Vining et al., 1983, and subsequent research, it is shown cortisol suppresses testosterone mechanism. Cortisol inhibits the hypothalamus, reduces GnRH, reduces LH. Additionally, cortisol and testosterone compete for resources within steroidogenesis. The body chooses either survival, cortisol, or reproduction, testosterone. Under chronic stress, cortisol always takes priority. Sources of chronic stress: sleep deprivation, overtraining, caloric deficit, psychological pressure, inflammation. All of them lead to one outcome: suppression of the HPG axis. Low physical activity, lack of movement also affects testosterone. Mechanisms: reduced muscle activity, reduced stimulation of androgen receptors, development of insulin resistance, fat accumulation, increased aromatase. This is an indirect but persistent factor in lowering testosterone. Alcohol directly affects the hormonal system. Mechanisms: Toxic effect on Leydig cells, reduced activity of steroidogenic enzymes, increased estrogen levels, disruption of hypothalamic function as a result. Testosterone production decreases. Deficiencies: zinc, vitamin D, Prasad et al., 1996. Zinc deficiency reduces testosterone mechanism. Disruption of synthesis enzymes, reduced LH, impaired Leydig cell function, Pills et al., 2011. Low vitamin D is associated with low testosterone mechanism. Reduced sensitivity of Leydig cells to LH. Final: testosterone does not drop randomly. It drops when the body receives signals of stress, energy deficiency, defeat, inflammation, and response as a survival system. It does not break, it adapts. And this is exactly what you need to understand if you want to truly influence it. Now the key question: if testosterone drops because the system is suppressed, how can it actually be increased? The most important point: testosterone is not directly boosted. It increases when the signals that suppress the system are removed and when the body receives signals of safety, energy, and adaptation. Fat loss, one of the strongest factors. In Ku et al., 2013, weight loss in overweight and obese men led to a significant increase in testosterone levels. But the mechanism is not just about weight. Adipose tissue is hormonally active. It contains aromatase, an enzyme that converts testosterone into estradiol. When fat mass is high, aromatase activity is elevated, more testosterone is converted into estrogen. Estradiol rises, estradiol feeds back to the hypothalamus and suppresses GnRH secretion. This reduces LH and lowers testosterone production when fat mass decreases. Aromatase activity decreases, estradiol levels drop. The hypothalamus is released from suppression. GnRH pulses become stronger. LH increases, Leydig cells receive a stronger signal. Testosterone production increases additionally. Fat loss reduces inflammatory cytokines such as TNF-alpha and IL-6. These cytokines are known to impair hypothalamic signaling and receptor sensitivity. Insulin sensitivity improves. This reduces chronic metabolic stress and lowers cortisol load, so fat loss does not just increase testosterone. It removes multiple layers of suppression at once. Resistance training, neural plus hormonal effect. In Kramer et al., 1990, heavy resistance training was shown to increase testosterone. But the key mechanism is deeper than a simple spike. During heavy compound movements, large muscle groups are activated, motor neurons fire at high intensity, the central nervous system is highly engaged. This leads to increased catecholamines, adrenaline and noradrenaline. These signals reach the hypothalamus. The brain interprets this as high physical demand, adaptation required as a result. GnRH secretion increases, LH increases, testosterone production increases, but one of the most important effects is often missed. Androgen receptor sensitivity increases. Studies show that resistance training upregulates androgen receptor density. This means the same amount of testosterone produces a stronger biological effect, so the system becomes more efficient. High-intensity training, sprints, and explosive work. In Trimbley et al., 1994, and later studies. Short duration, high-intensity efforts were shown to produce a strong hormonal response. Mechanism: rapid activation of the nervous system, increase in dopamine, strong activation of the sympathetic system. This creates a powerful adaptive signal. The body responds by increasing anabolic signaling, including testosterone. However, the key variable is duration. Short and intense positive adaptation. Long and excessive chronic stress if training becomes excessive. Cortisol remains elevated, HPG axis is suppressed, testosterone decreases. This is why elite endurance athletes often show low testosterone levels. Stress reduction, removing the main suppressor. Chronic stress is one of the primary blockers. If cortisol remains high, the hypothalamus is continuously suppressed. In Tang et al., 2007, meditation significantly reduced cortisol levels. In Jareth et al., 2015. Slow breathing reduced activity of the stress system mechanism. Reduced activation of the HPA axis, lower cortisol output. This removes inhibition from the hypothalamus. GnRH secretion normalizes LH, increases testosterone production, recovers additionally. Lower cortisol reduces competition in steroid hormone synthesis. The body shifts from survival mode So recovery and growth mode practical. Implication: the absence of chronic stress is not optional. It is required. Sleep, restoring hormonal rhythm. In Lepold and Van Cotter, 2011. Restoring normal sleep duration restores testosterone levels. Mechanism: testosterone secretion is pulsatile and tightly linked to sleep architecture during deep sleep. GnRH pulse amplitude increases LH pulses, increase testosterone peaks with adequate sleep. This rhythm is restored additionally. Cortisol decreases especially in the evening. This removes inhibitory signals from the hypothalamus. Sleep is not just recovery. It is the central regulator of hormonal timing. Dietary fats, substrate plus signaling. Involek et al., 1997. Low-fat diets were associated with lower testosterone. Mechanism is both structural and hormonal. Testosterone is synthesized from cholesterol. If dietary fat intake is too low, cholesterol availability decreases. Substrate for steroidogenesis is limited additionally. Cell membranes require lipids. Hormone receptors are embedded in these membranes. Low-fat intake can impair receptor function and signal transmission. Types of fats matter. Saturated fats support steroid hormone production. Monounsaturated fats improve metabolic health. Omega-3 fatty acids reduce inflammation and improve insulin sensitivity. Together, they create an environment where hormonal signaling is optimal. Zinc, critical cofactor. In Prasad et al., 1996. Zinc deficiency significantly reduced testosterone. Mechanism: zinc is required for multiple enzymes involved in steroidogenesis. It also supports LH production, and Leydig cell function deficiency leads to reduced enzymatic activity, lower LH, impaired testosterone synthesis. Restoration of zinc levels restores enzymatic pathways, improves hormonal output. Important point: this is not boosting testosterone, this is correcting a deficiency that was limiting production. Sunlight and vitamin D, hormonal regulator. In Pills et al., 2011, vitamin D levels were positively associated with testosterone levels. Mechanism: vitamin D acts like a hormone. It influences gene expression in Leydig cells. It improves sensitivity to LH deficiency, reduces cellular responsiveness, and lowers testosterone production. Sunlight exposure triggers natural vitamin D synthesis. This is a regulatory signal that the body uses to adjust hormonal function. Glucose stability, metabolic control. Coronia et al., 2013 showed acute glucose intake reduces testosterone by 25%. Mechanism is not only acute. Chronic instability in blood glucose leads to insulin resistance, increases inflammation, increases fat accumulation. All of these suppress the HPG axis. Stable glucose reduces metabolic stress, improves insulin signaling, reduces inflammation. This indirectly supports testosterone production. Final testosterone does not increase from a single action. It increases when the hypothalamus is no longer suppressed, cortisol is reduced, estradiol is controlled. Metabolic function is restored, receptor sensitivity is improved. This is not a shortcut. It is a systemic adaptation. The body does not boost testosterone. It allows it when conditions are right. Here, you'll get information that is rarely explained clearly and honestly. I constantly analyze scientific studies, meta-analyses, and real data. Everything related to muscle growth, fat loss, supplements, and nutrition. No guesses, no marketing hype, only what actually works. If you found this video useful, support the channel by liking and subscribing. And leave a comment with the topic you want me to break down next. Here we go deeper than most. We break myths and focus only on what delivers real results. Join us today for more facts and results.